Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So today we are going to learn about cytoskeleton. So cytoskeleton has two words. The cyto means cell and skeleton means the basic framework, which is going to help in maintaining the structure of that cell. Okay, so the cytoskeletal system is a complex system. It is a complex network of protein filaments that extends throughout the cell. So the way our skeletal system, the body skeletal system, helps to maintain the structural integrity of our body as well as it helps in the movement of our body. So, due to the bones, due to this skeletal system, we are able to move from one place to another. In the same way, the cytoskeletal system that is made up of different proteins, the different filaments, that helps to maintain the structural structure of the cell as well as the physical robustness of the cell, and that also helps in the cell movements. So it also helps in cell polarity. So today this is an introductory lecture where we are going to explore what cytoskeleton is, how it is made up of different protein filaments, and what are the different functions that it performs. So it is a dynamic framework which has different types of filaments. So cytoskeleton system is made up of filaments, three different types of filaments. The first ones are known as actin filaments. These are also known as microfilaments. The other type of filaments are known as intermediate filaments. And the third type is known as microtubules. Okay, so these different filaments, they have different roles that they perform in the cell. For example, the actin filaments, they help in muscle structure. They build the structural unit of muscles, which is known as the sarcomere, the structural and functional unit. Then again, intermediate filaments, they form the laminar structure, the nuclear lamins in the nucleus. The microtubules, they form the different motile structures like cilia and flagella. So we will be learning about those structures in upcoming lectures. So those different types of filaments, they form different structures which perform different functions, but together they give the cell polarity. They help the cell to maintain its shape, and they help in the cell movement. So the way our muscles, the bones, ligaments, and tendons, they come together and they help maintaining the structural integrity of body. In the same way, those different types of filaments, they work together, they do their own job, their different functions, and then they help the cell to maintain its structural integrity. So this is a dynamic framework. I'm calling it a dynamic because it constantly assembles and disassembles, responding to cell needs. So these three types of cytoskeletal filaments, they are important. So they are made up of they have some subunits, means they are made up of a protein monomer. For example, the actin filaments, they are made up of the globular monomer known as G-actin. So this protein polymerizes to form actin filament. Okay. Then the intermediate filament, that is made up of long proteins known as intermediate proteins. So the intermediate proteins of different types. So like there are different types of intermediate proteins like vimetins, the keratins, the acidic and basic keratins. So they come together to form intermediate filaments. Then there are tubulin proteins when monomerized, they form microtubules. So tubulin proteins like alpha and beta. So then there are some minor tubulin protein types like gamma, delta. They come together to form microtubules. So these are the monomeric subunits which polymerizes to form those filaments. So how those filaments form different structures. So for example, the cells which have fixed cell shape, for example, the neurons or the epithelial cells. In those cells, the cytoskeleton builds and maintains large stable structure. The example is microvilli and cilia. So the microvilli, these are the finger-like projections that are found on the gut, which helps in the nutrient absorption in the intestine. So this microvilli increases the surface area. So this is maintained. This structure is formed by the filaments, those cytoskeletal filaments. Then cilia, which are found in respiratory tract and in the fallopian tube, which helps in the sensing environment as well as movement of the substance in certain direction. They are also made up of the cytoskeletal elements. So those protrusions, they are uniform in size and shape, and even in the location is constant throughout the life of the cell. So that is a very interesting thing, that how the shape is being maintained, the structure, that specific structure is being maintained throughout the life of that cell, or throughout the life of our throughout the life of an individual. So now there's one more example of such structures. So we discussed microvilli, we discussed cilia. So there's one more structure known as stereocilia. Okay, stereocilia. So these are the inner ear hair cell, inner ear hair cells. Okay, so these are also the structures that are formed by the cytoskeletal elements. So those stereocilia, these are specialized actin-based structures. Okay, so these are actin-based structures. Okay, so they are very interesting, because this precise architecture is maintained throughout the lifetime. Like we live a long life, and throughout this lifetime, this hair cells, those this structure is maintained, and this actin filaments, they are not stable. I mean, they're dynamic. The polymerization and depolymerization of the tip of those hair cells is seen throughout the lifetime. But this is how they are maintained throughout the lifetime at such constant length for so long. That is one of the interesting questions, and one of the big mysteries in the biology. So this is how the different kinds of big structures are formed by the cytoskeletal system. Then cytoskeletal system also maintains the cell polarity. Okay, so more than supporting structure, it also has function in maintaining or setting up the polarity. So it helps the cells to distinguish from between the top and bottom, front and back. So polarity of the cells is very important, and this is a crucial function which is performed by the cytoskeletal system. So for example, epithelial cells, these are also polarized cells. Okay, means they have a sense of directionality. So, for instance, the organized arrays of actin filaments or microtubules and intermediate filaments, they preserve the differences between apical and the basolateral domains. So those cells, the epithelial cells, they form different types of junctions, like tight junctions, and they form the effective barriers in the intestine and lungs. So having the polarity in the epithelial cell is a very important characteristic, because it is going to result into a lot of functions. If the epithelial cells loses its polarity, and it kind of have transition from the epithelial to mesenchymal type of cell, so this could lead to cancer. So how the cell polarity is maintained, and how the different types of filaments, they give the cell its polarity, the sense of the directionality, we'll be discussing that in upcoming lectures. Today we are just discussing what kind of functions are performed by the cytoskeletal system. So the very important function is that it maintains the cell integrity. Okay, so it maintains cell shape, structural integrity. The physical robustness. Okay, and it also maintains cell polarity. It forms the specific structures like microvilli, cilia, stereocilia, which has different types of functions in the cell. Okay, so cytoskeletal system, it also, is dynamic. That also we learned in today's lecture. So let me show you a diagram of all those three different filaments. Okay, so all of those diagrams I've taken those from a book known as Bruce Albert's. So if you want to read about this particular topic, cytoskeleton, you can refer this book. It's a very nice book, nicely narrated. Okay, so this this picture, this is the single actin filament, okay, which are also known as microfilaments. So the actin filaments, they are from 7 to 8 nanometer in diameter, okay, and they help in the maintaining cell shape. So the actin filaments, they are usually found in the cortex of the cell. So cortex is the cytoplasmic area which is present just beneath the plasma membrane. So let's say that this is the cell, okay, and that is the cell membrane. So the cytoplasmic area here is known as cortex. So in this area, so this was a 2D diagrams, but imagine this in a 3D way. Imagine that it's a ball, okay? So the area of the cytoplasm, which is just beneath the plasma membrane, is known as cortex. So here the actin filaments are found, and this is how the concentrated actin filaments, which are found in cortex, they help the cell to maintain its shape. So the 3D shape of cell is maintained due to the actin filaments. So actin filaments form certain structures such as microvilli. So this is the diagram of microvilli, okay, and here you can see the actin filaments which are stained, okay, most probably it's phalloidin. Okay, so these are the actin filaments which are being stained. And again, the very important function of actin filament is that they form the structural and functional unit of the muscles, which we call as the sarcomere. So this is the this is the image of the striated muscle, the skeletal muscle. Here you can see the actin filaments. Okay, so we'll discuss about this in detail in upcoming lectures. This is the diagram of microtubules. So microtubules, they are long and hollow cylinders. So unlike the actin filaments, which are which are not hollow, the microtubules are hollow structures. So here you can see the diameter, the outer diameter of those structure is 25 nanometer, while the inner diameter is 14 nanometer. Okay, so this is an hollow structure, and it is very important because it is going to form the centrosome, it is going to form the motile structures such as cilia. So here you can see they are showing the cilia, the triplet structure of the cilia. We will learn about the structures of cilia and flagella in upcoming lectures. So I'm just introducing to the basic idea to you all. So here this image is the interface of the cell cycle, and here you can see the arrangement of microtubules. The green, those are the microtubules, and these are the organelles. Okay, and here you can see the image of a ciliated protozoan. So these are the cilia. The green structures are the cilia, which are made up of the microtubules. And this figure is a single microtubule. So this is how a single microtubule looks like. Okay, so this monomeric proteins, those globular proteins, are known as tubulin proteins. Okay, so the microtubules are made up of tubulin proteins. Here you can see intermediate filaments. So intermediate filaments, they're less dynamic compared to the actin or the microtubules. The diameter of intermediate filament is 10 nanometer. So they are called intermediate again because the size is somewhat intermediate between both of the filaments. The actin filaments have 7 to 8 nanometer diameter. Okay, the intermediate filaments have 10 nanometer diameter, while the microtubules have 25 nanometer diameter. So the size is the intermediate of intermediate. It is in between both of the filaments, and that is why the name intermediate filaments. So they are made up of intermediate proteins of different kinds. We will discuss that in that particular lecture. We'll be discussing how the those different proteins assemble to form intermediate filaments. So here you can see the diagram showing the schematic showing the intermediate filaments. So they are rod-like structure. So they are not hollow. So microtubules are hollow structure, but intermediate filaments are rod-like or rope-like structure. And it is the heterogeneous filaments, the long intermediate proteins, they assemble together. And intermediate filaments, they are often found in epithelial cells. They are found, and the very important place there where you will be finding the intermediate filaments is the nuclear lamina. So in the nucleus, just beside the nuclear membrane, a mesh-like structure is formed, which is very important because it is going to maintain the shape of nucleus, as well as it is it is important to hold the chromatin in its place. So this structure, the mesh-like structure, is known as nuclear lamina, and this nuclear lamina is formed by the intermediate filaments. Intermediate filaments can also be found in neurons. So this is the image where of the intermediate filaments, how they are assembled in neurons. Okay, and this is the image of an epithelial cell and the arrangement of intermediate filaments in the epithelial cells. So this is how the intermediate filaments, they form a mesh-like structure. Okay, in the nuclear lamina. So this is the new image of an nuclear lamina. So with this, I end this lecture. I conclude this lecture. In next lecture, we will learn about certain motor the accessory proteins of the cytoskeletal system. We'll also learn about the actin filaments in detail, how the G actin, the monomer, gets polymerized to form filamentous actin, and the process of the actin treadmilling, the filament treadmilling. We'll learn about all those stuff in next lecture. So after this, we are going to learn about all of those filaments in detail, one by one. Till then, see you. Bye bye.